He's a director of the Center for Membrane Applications, Science and Technology, an industry university cooperative research center. He obtained his bachelor's and master's degree from the University of Melbourne and his PhD from the University of Minnesota. He worked in the biotechnology, biomedical industry in the Boston area before joining the Department of Chemical Engineering at Colorado State University. He joined the University of Arkansas in 2011. So, yeah, please.
Should I use the?
Yes.
Okay.
Sorry. I can about this one.
Yep.
And,
Oh, I can.
yeah. Please join me in welcoming Professor Rani.
Thank you very much. So thank you very much for the introduction. Also, thank you, Sean, for the invitation. I tried to visit once before, I remember during COVID, and it didn't quite work out. So it's good to be able to visit UTS. I chose a topic that's a little bit different perhaps to what you do, and so please feel free to interrupt me because I'm going to talk about bioseparations, and maybe there'll be some interesting questions and so forth, so please feel free to ask me about it. The other point is I extended the talk, so the talk is actually longer than 30 minutes. What I can do is skip parts and so forth. I hope that's better, and you'll see how it goes, okay? So, this is what I was initially trying to do, talk about membrane challenges in bioseparations, and there are 3 different challenges I was going to talk about, but we can easily skip one of these. Today, mainly batch processing, continuous biomanufacturing, and virus purification. And I was going to talk to you a little bit about some of the work we do with membrane distillation for treating hydraulic fracturing produced water. So, the first point is that we have 2 membrane centers. One is called the Membranes Application Science and Technology Center. It's a collaborative center. University of Arkansas leads it. It's with the University of Colorado Boulder, New Jersey Institute of Technology, and Penn State University. And our themes are basically water purification, biopharmaceutical processing, and then some others here. But biopharmaceutical processing is rather a big theme in the center. The center is one of these NSF Industry University Cooperative Research Centers, and the idea, and this is an NSF diagram, but the idea is that NSF tries to get universities to work with industry. Industry really provides the industrially relevant research problems, that's important, as well as the funding. The universities provide the expertise, the professors, and also the graduate students, and also, of course, the equipment. And the whole idea is workforce development. So we're working with PhD students who will actually end up working in industry. That's the main focus of this. And we lead the development of new technologies. I thought Professor Ho's talk was very interesting because we fit in this gap here. So he's talking about commercialization, and we are supposed to fit in this gap here. So this is fundamental research, and then there is this area. And that's important because I told you that our work is funded by industry. These are some of our industrial sponsors, and what you see is firstly, of course, by the biotech companies, right? Like AbbVie, CSL Behring, AstraZeneca, Biogen, etc. But also you have membrane companies, so you can see where's Millipore Sigma, Paul Corporation, now Cytiva, and so forth. Repligen and so forth. And so the idea is that if you look at this, you can see there are many competitors who are working together. That is where that gap is very important, the sort of research we do. We cannot work on research that's going to enable one company to get advantage of another one because they'll never join the center. What's the point, right? But what we do is work where the users of membranes can work with membrane manufacturers on problems that are of general interest, like I was mentioning membrane fouling. And this is a general interest to all the biotech companies. So if we can find a topic like this, it can't be highly theoretical. It has to be of applied, of industrial relevance. But that's the sort of space we are working in. Okay? We also have some others, Bureau of Reclamation for water treatment areas, and the dairies, food industry, and so forth. Okay. The other center we have is called MVP, Membranes for Virus Purification. This is entirely funded by NSF. It's this EPSCOR program, and it's a collaboration between University of Arkansas, which leads it, Clemson University, and University of Kentucky. It's interesting because this center is focused entirely on using membranes for purifying viruses, virus vectors, if you like, particles. And there's a great deal of interest in these sort of developing therapeutics based on viruses. So we look at things like feedstock production, bioreactor harvesting, affinity separations. I'll talk a little bit about full and empty capsids and so forth. And so this center these are entirely research problems that come out of the faculty, not industry as such, but they have industrial relevance, of course. Okay, so moving on to this idea of bioseparations. So please stop me if you have questions. If we think of bioseparations, you can divide the manufacturing process into two stages, so-called upstream processing and downstream processing. So in the upstream processing, we're going to use cells, typically mammalian cells, to produce our product of interest. So we have various bioreactors where we increase the size of the bioreactor, where we're increasing the number of cells. At some point, we switch over and we get these cells to now produce a product of interest. In so-called traditional biotherapeutics, so I would call them monoclonal antibodies, protein-based biotherapeutics, these protein of interest is excreted by the cells into the growth medium. So it's extracellular. And then what happens is we have these what are called midstream operations, basically bioreactor harvesting or clarification operations, where you remove all the particulate matter, you just want the liquid phase. And that's what goes into the downstream purification platform, which is what we're talking about here. Now the problem today is that, of course, these monoclonal antibodies are very expensive, there's a great demand for them, but the bottleneck in the manufacture of them actually is in the downstream purification part, not the upstream processes. So upstream processes have improved, we can grow cells to high titer, we can produce a large amount of protein, but we have difficulty purifying it effectively. So that is where really the problem is. The other point is that the biotech industry is highly regulated, right? If you make any change, excuse me, to make any change, you're going to have to get approval again from a regulatory agency, FDA or something else. This is very costly and expensive. So the main focus in industry is to take existing unit operations and can you improve their performance? That's really what the focus is. So as I said, we're using mammalian cells. We use them to produce monoclonal antibodies. Chinese hamster ovary cells are typically used. Now I'm going to focus on virus removal and virus purification, since that's an interesting area and something that we work with. So these mammalian cells produce virus particles. They naturally produce non-infective, what are called retrovirus-like particles. So you have to show that they are removed from your product. You're not going to give someone a product which is contaminated with these virus particles. They may be non-infective, but they can still cause very serious side effects. So this is a very, very serious problem. It's in fact one of the reasons why gene therapy applications have taken so long to develop. It's been very difficult to actually purify the product. So there are FDA guidelines that ensure adequate virus clearance. You've got to show that in your purification train you would have achieved the amount of virus clearance that could theoretically be possible. It doesn't mean it exists, okay, but it could be possible. Now there's another type of virus clearance that also is important, that's adventitious virus clearance. So it also is possible that by using cell culture derived growth medium, that viruses from the growth medium are somehow survive and get into your product. Another reason for adventitious virus clearance is that, we always want to make sure that our product is safe, and we are discovering new viruses all the time. So the best we can do is, we work with a model that's particularly difficult to remove. So by today's standards, we know if there's anything else there, it will be removed. Of course, that model becomes outdated if we find a virus that is more difficult to remove, okay? And removal is either by inactivation or by clearance, okay? That's how you remove the virus. We focus on virus filtration, which is a robust method for virus clearance that rejects virus particles from the process stream based on size. So you're going to reject the virus particles that the product of interest go through. This is a very difficult separation because the virus particles, the smallest ones, are maybe 20, 22 nanometers in size. The largest monoclonal antibodies could be about 10 nanometers in 100 nanometer radius. That's a very tight separation. And so virus filters are typically large core ultrafiltration membranes. Before I talk about that, so here's the downstream purification platform in more detail. And so just in case, because you probably may not know that much about it, you get your feed here, your clarifying feed, it comes in. Typically there's a so-called capture step, protein A chromatography, and there are 2 polishing chromatography steps. That's typically what one would find. One thing to remember in the biotech industry, all packed bed processes are called chromatography, okay? So we might call these adsorption, but they call them chromatography. This is called bind and elute chromatography. Protein A is the ligand. You bind your product of interest and you elute. Polishing steps typically are what are called flow-through chromatography in the biotech industry. It means that you're binding the contaminants. The product of interest just flows through the column. Okay, that's typically what that means. So if we look at this, besides that, there are many other things that occur. You can see a pH adjustment vessel here. Typically, one of these polishing steps is also involved, and virus filter is involved. All three are typically used to validate virus clearance, because one process by itself would not be adequate, because the level of clearance you have to show is too much, okay? And there are reasons for that which we could discuss. And so pH adjustment means you will reduce the pH to a lower value, about 3.5, something like this. If the product is stable, that inactivates retroviruses. Polishing chromatography typically use anion exchange membranes. You can absorb the contaminant, absorb the virus particles, the monoclonal antibody flows through. You need a different isoelectric point to make that work. But we are talking about virus filtration. So these are very, if you like, large pore ultrafiltration membranes. But look at this, the virus filtration step occurs very close to the end of the purification train. There's one ultrafiltration, diafiltration step typically after that. And that is used to concentrate the product and put it in the buffer of interest. So by this stage, the product should be very pure. It's at the end of this very complicated and expensive purification process, okay? So one of the things that comes up then is that virus filters are made to very, very tight constraints. You cannot have any macro defects because you want to show at least maybe 10,000 times or 10 to the 4 log clearance of virus, so 4 log clearance. So you want 10,000 times reduction in the virus load in the feed compared to the permeate. You also want at least 90-95% product recovery. So that's very, very tight and very difficult to do. And so these virus filtration membranes, just remember, they are running in opposite to ultrafiltration membranes. The support structure, the asymmetric membrane, the support structure faces the feed stream, and it acts as an inline pre-filter. They're always run in normal flow or dead-end mode. Okay. And as I said, it occurs close to in the purification train. So fouling, therefore, cannot really be due to contaminants. Mainly, it's going to be due to product-related contaminants, aggregates, associated protein, misfolded protein. So they are product-derived contaminants typically. So one study I thought I'd show you quickly, and this is for batch processing, which I showed you earlier, is that you saw at the very end, the virus filtration occurs at the end, but then there's an ultrafiltration diafiltration step. It'd be nice to eliminate that UFDF step. Why can't we just run through the virus filter? After all, it is a very tight pore size membrane. What you get out of the virus filter is very pure. Why can't we run that directly into formulation? The problem is that you want a higher protein concentration. These virus filters typically don't work very well at high protein concentration. They foul. And secondly, you may have to change the buffer. So we did some work where we worked with a monoclonal antibody from one of our sponsors, and we looked at different concentrations. So today, typically virus filtration run at about 10 grams per liter of protein. And you can see, if you look at the flux versus throughput in meter per meter squared, permeate volume per membrane surface area squared, if you increase the protein concentration, the fouling becomes much more significant, and this flux goes to zero very quickly. If you plot this versus mass throughput, the amount of protein you recover in the permeate, you see them at about the same point. So fouling is occurring, but really it seems to depend very strongly on protein concentration. So then, to cut a long story short, we looked at various ways to try and understand the protein. We looked at size exclusion chromatography. We looked at various other things. And so SEC analysis suggested that there really weren't that many aggregates present. So that wasn't the problem. But we looked at the hydrodynamic radius of this protein in the same buffer at different concentrations. And what you see is that if you look at higher feed concentrations, so say something like 47.8, that's the green curve, you get this sort of a curve here for the protein hydrodynamic radius as given by dynamic light scattering. But if you look at a lower concentration, 7.2, you get a much sharper peak at a lower value. So the protein hydrodynamic radius cannot change. But what it means is that at higher concentration, you're getting some sort of loose association of this. And that is what is then going and causing fouling of the membrane. So one way around it is to put a pre-filter. I'll just talk about this one, an ion exchange, a cation exchange pre-filter, and you see you get much more stable fluxes. Now, what this is though, I told you can also use anion exchange chromatography as a virus clearance step, but this is a pre-filter in line with your virus filter. There cannot be any gap or anything in between, and that's one way to overcome this. So that's one of the challenges that one looks at in this area of current batch type processing, and there are others which we could talk about as well. But pre-filtration is an area that is quite important. There are many types of commercial pre-filters available based on ion exchange, hydrophobic interaction, and so forth. Okay. Let me move on. If you have questions, please stop me. But let me move on, because one of the emerging trends in biomanufacturing is something called continuous biomanufacturing. So, how many people here are chemical engineers versus civil engineers? Chemical engineers. Okay. So for chemical engineers, continuous, we typically think of a chemical plant, and you're running a cat cracker, I don't know, for a year or more, right? In the biotech industry, continuous biomanufacturing would mean maybe 1 month, maybe 2 months. But it's certainly not 8 hours or something like this, okay? Because at the moment, you grow your cells, that takes a while, and then each unit operation should not take more than 1 shift, at most. But for continuous biomanufacturing, there's a great deal of interest. Firstly, it reduces cost, right? Because you don't have all this equipment downtime. That's well known. And secondly, there's more flexibility, because you run enough to produce enough product for several years, and then you can switch to something else, right? So that also helps. The other very important thing is that you're running under steady state conditions. Actually, this is a very important point, because in the biotech industry, it's a biological system, so any slight variations do lead to product variability. If you can run under steady state conditions, you have much better control of your product. So, if we look at how, and of course it leads to reduced glycosylation, deamidation, and so forth. So if you look at this, here's a perfusion bioreactor. The idea is that you have your clarification steps. So you're continuously removing some of the liquid and putting the cells back. You do this continuously, right? But then you have your primary capture step, as I showed you, protein A, polishing steps, formulation. All of these now need to be continuous. So this becomes quite a challenge, okay? So other investigators have looked at the following. They've looked at trying to develop continuous tangential flow microfiltration. So if you think of microfiltration, it's a batch process, right? You have your feed tank, you have your retentive tank, you keep recycling as much as you need to concentrate or diafilter, whatever you want to do. But the idea with continuous SPTFF, single pass tangential flow filtration, the feed comes in, and this could be one module or it could be multiple modules, but the flow paths are 3, then 2, then 2, then 1 for the feed. So as you're removing permeate, you reduce the feed flow paths to ensure the same wall shear stress. So you should be therefore running at essentially steady state conditions. But the important point is it's just one pass through this. You don't have constant recycling going through the feed pump and so forth. In the case of diafiltration, one can do that also. And so the idea with continuous diafiltration, this actually comes from gas separation. So you can see the feed comes into the first module, then the retentate becomes the feed to the second, the retentate becomes the feed to the third. The diluent buffer enters here, the last module, and then it goes countercurrent to the feed. So by countercurrent contacting, you can actually show you require less diluent. Okay, but this is a continuous process. The feed is going through each of these modules only once. Okay, it's not recycling. The question is, so that's fine. The question, and by the way, you can buy SPTFF units now commercially. But the question is, how do you make virus filtration continuous? So I told you about virus filtration. We're going to run the product through. We want a certain amount of virus clearance, and we want to validate that. This becomes a very difficult problem. There are many complications, and whether you can make it truly continuous is something that, if you like, we can discuss. But the important point is that virus filters today are run at constant pressure to maximize the flux, because you want to get it done in one shift. If you want to look at continuous biomanufacturing, this needs to now connect up with, say, a chromatography step or something else, so that the flow through from that needs to go continuously through the virus filter. So you're going to run at much lower flow rates, but you want to run at constant flux or constant flow rate. That is the critically important point here. So it seems that this might be easy, right? You take current virus filter, you run it at a constant flux, and that should be easy. Unfortunately, it doesn't work out quite as well as that, and so that's what we talked about here. So we worked with proteins from Biopharmaceuticals. These are the buffer conditions. We did use a pre-filter. I explained about pre-filters earlier. And the virus we used is minute virus of mice. It's a FDA recommended parvovirus. It's typically used to validate adventitious virus clearance. We grow it ourselves and we purify it to a very high level, and we use it in our spike studies. Now, if we think about this, what happens is, if you look at the current fluxes under which virus filters are run, they are in this so-called convection-dominated regime. So if you look at the Peclet number, which is one of these dimensionless numbers, it's a ratio, if you like, of convective flow to diffusive flow. So at current fluxes that we run, typically convection dominates. And the idea here is that you have a virus filter, think of a asymmetric membrane, right? So you've got through pores, but there's tortuous pores, there are constrictions. The virus particles come and get stuck, that's what you hope, in these constrictions, and stay there, they don't move. And so you have a strong convective force that's pushing, and that keeps them there. But if you want to look at continuous biomanufacturing, you're going to run much lower fluxes where the Peclet number actually can now be less than 1. And so diffusion dominates. So it's possible that these virus particles come, but then they can also back diffuse and then find a larger pore to go through, and so you could have breakthrough. And that is something that is already observed in batch processing. I'll explain why later on. So the idea of this study was then to look at commercially available virus filters and operate them under these different conditions, okay? Under conditions where they are rated to work, and then under conditions where we have Peclet numbers around 1 and Peclet numbers much less than 1. These are the filters we used. We label them like this, but if you know anything about the filters, you can guess who makes them. But these are hollow fibers. We generate cellulose. This is hydrophilized PVDF hollow fibers, polyether sulfone flat sheet, and PES again hollow fibers. Okay. One thing to note here, this is the again for chemical engineers. Usually we scale up. Here we're scaling down, right? You have a manufacturing process. There's no way I'm going to spike that with virus and see what happens. You need to scale down to a very small level, but this needs to represent the large-scale process. So you're going backwards. Okay. So therefore, the membrane surface area is important, and then the permeate fluxes are important, and the throughput, or the amount that you produce, the amount of permeate you recover is also important. Okay. So I'm again going to skip the details and present you with the results. So this is a virus titer in various fractions. So we run this in dead-end mode, and we collect our virus in the permeate in various fractions. Fraction 1, fraction 2, fraction 3. Each of these fractions actually is about 50 ml. And these fractions are worked out so that every filter, which has a slightly different membrane surface area because manufacturers make them with different surface areas, will be subjected to the same throughput. Okay, you have the same amount of permeate collected per membrane surface area, normalized by membrane surface area. So this is filter V, filter N, and filter B. This is a virus titer in each of these fractions measured by something called PCID50 assay. It's an infectivity assay. It measures how many virus particles are there. So look at this one. So this one, you can see that we have maybe about, this is in log scale, so say 1.95, 2 point something, and you can see that you can measure all these values, and you can see the virus concentration in these various fractions, 1, 2, 3. BC is something called buffer chase. So that's what I mentioned earlier. I told you there's this hypothesis that virus particles get stuck and could back diffuse. Well, this has kind of been observed. So in current manufacturing, you're running the virus filter, you treat the whole batch, and then the product is quite important, so it's very valuable. So you flush the system with buffer. In doing that, there's a pressure drop and then a pressure rise again, right? Because you've got to switch over. And it's noticed often that you get virus breakthrough during this buffer chase. And so we did that with manufacturing, and you can see here the virus in the buffer chase. So what this shows you in all cases except for this one, we do see virus particles in these various fractions. The initial titer was 5.5 log. We expect to get at least 4 log clearance, so these are showing less than 4 log clearance, except in this case. This filter was actually pretty good. And so we're getting breakthrough. But if we run at higher fluxes, this is at 40 LMH, you can see the exception of filter N, none of them show any breakthrough, all below level of detection. So in the case of continuous biomanufacturing, one big problem is how you design these virus filters. Okay, we actually got quite interested in this and looked at something else. So some other things that occur in this whole area that are interesting is that if you look at the operation of a virus filter, it's a little bit different to say other sorts of membrane processes, in that if you get fouling, you sometimes get worse performance. You get virus breakthrough, which is odd, right? If you get fouling, you expect the flux to go down, but you expect not to get a drop in rejection, but you often do. And so what we did is, and I'm sorry the scale seems to have got messed up here, but what we did is we took, again, a monoclonal antibody in our feed solution. There's no virus here this time. start again. We took one of our virus samples and we spiked it with either PBS or this monoclonal antibody at 5 grams per liter. So it's a low concentration. In the case of PBS, you can see, if you do SEC analysis, that there's quite a lot of dimer present, that's some sort of aggregate, and trimers. In the case of the monoclonal antibody, there's almost nothing, okay? It's almost all pure monomer, okay? So the question is, where do the virus particles get stuck in the membrane? We want to see that, and does the fact that we use these different products in there cause a difference? That's what we want to see. So I just show you, and I have many more of these results which I can show you. So these are two different Asahi modules, 20N and S20N. They are both regenerated cellulose membranes. And I don't know why the scales got messed up again, but anyway, if you look at it, this is MVM only, virus only. You get this red curve. It's based on these confocal microscopy images, where this is the inside surface of a hollow fiber, this is the outside surface, and so the permeate is flowing in this direction. So this is where the virus particles are being trapped. These are labeled MVM virus particles, okay? And based on that, we can do this sort of analysis and find out the relative position, this being the inner surface, that being the outer surface. If we have a monoclonal antibody present, this red peak moves a little bit towards the outlet, but it's still very sharp. But if we use BSA, where we had many more aggregates present, the peak not only migrates towards the outlet, it also broadens, okay? And the same is observed with this other membrane. So this suggests that, firstly, I talked about the fact that you can get migration of where viruses get trapped if you reduce the pressure, right? You reduce the pressure, they can back diffuse and go back. But another thing that comes up again on this hypothesis of viruses getting stuck in constrictions is that if you have aggregates present, they are competing for these constrictions with the aggregate particles. And when they do that, then in fact some of them start to get trapped much, much further into the membrane, and there is a possibility of breakthrough. Again, explaining why sometimes when you have fouling of the membrane and dropping flux, you also see breakthrough of virus particles. So to end this part, then we tried to model this, and so we looked at using a log normal pore size distribution to model the pore size of the membrane. Remember, these are asymmetric membranes, okay? And then we also need to include a factor where we look at the projected area of a virus particle and the pore. Remembering that the virus and the pore, the virus we assume is approximately spherical, that's reasonable. The pore does not, it can be any shape. And so we need to account for this variation, okay? And then the other thing we did in this model, these are the main features of this model, is that we break up the membrane into a number of layers like this, each of which could have its own log normal pore size distribution, or a different one, because the membrane structure is changing, right, as you go through it. But importantly, there's a gap between them, because you wanted to try and numerically account for the fact that virus particles can back migrate and then move forwards, and in this way can be trapped in lower and lower layers. So it's not like virus particles of a certain size are all trapped in one layer and then the next layer and the next. There's a possibility of going back and forwards. So this work has been published. I could talk about it to you more later. What we're trying to do now is incorporate the idea of the interaction of aggregates in the product stream. Okay? Okay, let me keep going then. I have a little bit more time, I think. So let me just summarize this. This is a constant flux filtration was conducted on 4 commercial virus filters, and we found that actually at low fluxes, diffusion dominates, and this model of diffusion dominating seems to work very well. We also show that there is an effect of aggregates on the location of where virus particles are bound. And then we also are developing this model to try and describe this. I want to tell you about something else on the biotech area, which I think is interesting, and that's all removing virus particles because they're contaminants, right, in the manufacture of proteins. But you know, today there's a great deal of interest in newer therapeutics. So we look at cell-based therapies, we're looking at virus-based therapies. The idea is you're developing therapeutics for very specific locations. If you take a drug, if you swallow an aspirin or something like this, it goes all over your body, right? It's going to affect everything. And that's not very good sometimes. You'd like this to go specifically to a specific location. And so virus particles as vectors for taking a therapeutic are very interesting. Gene therapy applications, viral vectors, because they're very specific. They will go specifically and attack a specific cell. I should say that there are other ways to do this. People are also looking at polymeric carriers, okay? But these are biological carriers. So this is my attempt to try and look at a platform then for purifying these virus particles, which is what that center is looking at. But again, you have clarification, isolation. You do typically have this nuclease treatment, purification, polishing. This is a new one, and the reason is that unlike proteins, virus particles are often intracellular. So you have to lyse the cells to get the virus particles, which causes a big problem when you come to clarification. The other point is, if you look at this in the polishing steps, there's one of these interesting ones. It says remove remaining contaminants and defective virus particles. So the question is, what are defective virus particles? This does not exist in protein-based purification, okay? So any virus that's produced by natural means will not, the cell will not produce 100% virus particles that are infective, okay? There will be some defective particles. These are so-called empty capsids, and the ratio of empty to filled can be very high. It can be as high as 10 to 1. It depends on your cell culture process. These empty capsids or particles are naturally occurring contaminants. Their presence reduces the effectiveness of the therapeutic, because the therapeutic is going to go and bind to the virus particle, bind to the cell, but if you have a defective capsid, it's not going to do anything, right? It'll bind there and not do much at all. Interestingly, today, there is no regulatory guidelines on how many defective capsids one should remove, unlike virus clearance, but it's very likely to come up in the future, okay? And that's probably because these gene therapy applications are only just gaining as commercialized products, okay? So our thought was, can we find a way to remove these? So I should tell you, industrially today, there are a few gene therapy products. This is done by typically anion exchange chromatography, resin-based chromatography. It's not very effective because you're losing up to 50% of your product, which is not a very good thing. That's why some of these gene therapy regimens are very expensive. One reason why they're expensive. So our idea was to look at if we could use absorptive membranes for this separation. Maybe many of you know absorptive membranes are good because they overcome poor diffusional resistances. So this could be an ideal application. But the challenge is this, and so I'm really just going to talk about the challenge. Filled capsids, this is for our AAV. So adeno-associated virus is probably the leading candidate for gene therapy applications today, that and lentivirus, okay? It's a small virus, 18 to 24 nanometers in size, but if you look at the full capsid and empty capsids, it depends on the serotype, but the size is basically the same, okay? The isoelectric point will be different because the filled capsids have DNA inside, which is charged, so that will change the isoelectric point. It will also change the hydrophilicity, okay? It will change the density, and I should point out density is important because today microbiologists use density gradient centrifugation to separate them, but that is not scalable, okay? And so what we thought is maybe we can try and look at multimodal membranes where we can look at differences in isoelectric point and hydrophilicity to try and separate them, okay? And one very easy way to do it is you look at the ratio of UV 260 to 280 because the presence of DNA causes a shift in these peaks, you see. And so these ratios will tell you if what you have. You can also do it by infectivity, QPCR. Okay, so let me just give you a couple of quick things on this and then I want to finish off with water treatment. So we did some experiments. These are buffer conditions and so forth. We're going to use a gradient elution. So the elution is going from no salt, basically 0 to 100% buffer B. Buffer B contains 1000 millimolar sodium chloride. Buffer A contains only 125 millimolar. So we're eluting with an increasing salt gradient, okay? So here's an example of one of the ligands that we produced and attached to the membrane. And the point is that if you look at this, you can, this is the absorbance versus time. That's loading, washing, elution. So you can see we do get two elution peaks here, a very small peak and a very large peak. And we are able to somewhat separate these peaks, and we find that this second peak actually is much enriched in the filled capsids, and the other one is enriched in empty capsids. And so there are other slides I could show you on this. It may be possible to achieve this separation. The problem is, though, the peaks often overlap, right? And trying to get them to separate, therefore you can get efficient, is difficult. Because another thing I didn't tell you, it's not just filled and empty. It's filled, empty, and things in between, partially filled and so forth. Okay. So membrane adsorbers appear attractive. If you're interested, there are other methods people are looking at. They're trying to crystallize virus and do various things which we could talk about. let me just talk there for a few minutes. I'd like to also acknowledge funding and various people who have worked on this. But let me, in a few minutes, just tell you a little bit about what we've done in the water treatment area. So this is something we've worked on over the years, integrated electrocoagulation and membrane distillation for treating hydraulic fracture in produced water. And I'll just give you a summary of what we've done. So the motivation came from the fact that hydraulic fracturing is very big in the US, especially places like Texas, used to be big in Arkansas. And the problem is that you produce a very large amount of produced and co-produced water that is highly impaired. It is a very nice way to be able to recover oil and gas from unconventional resources. And so how do you dispose of this water is mainly the issue. And the level of treatment depends upon the use for the produced water. So the hydraulic fracturing operation basically is taking a drill and going to the Earth's crust until you reach this tight rock formation, and then turning it and going horizontally for up to 3 kilometers, and then you send water at high pressure, which basically fractures this rock formation. I mean, it fractures it, it causes fissures. You have proppant, which is basically sand particles that go and get stuck in these fissures. When you release the pressure, you get the flowback water, you get your oil or gas, but the rock is more permeable now, so you're more able to easily recover the oil and gas. Okay? Sorry. And so the problem is what you do with this water, because you're using water to fracture the rock, and now you have a very impaired source of water. And so today most of it is trucked, this is a schematic representation, and deep well injected. So there are a number of problems to this with deep well injection. Firstly, you are actually, it's a cost of transporting this all the way to a site. Secondly, you've got to make sure that the site that you're injecting into doesn't affect any other water body, because if it does, you're going to contaminate other groundwater that could be used for beneficial uses. It also causes earthquakes, which is known to happen, and it's not very sustainable, because if you think of it, you're taking water, a very valuable natural resource, and you're trying to get rid of it, right? You're trying to deepen the Earth's crust and get rid of it. And so there is a great deal of interest in how to treat this. So we worked over the years on various processes. Basically, we worked with colleagues at Texas Tech to come up with an electrocoagulation and membrane distillation step. So that would be taking produced water, we run electrocoagulation as a pre-treatment step, we then use ultrafiltration or microfiltration to separate the sludge we produce from electrocoagulation. The permeate from that then goes to a membrane distillation step. And at Texas Tech, they were using VMD, vacuum membrane distillation. So we worked mainly on the electrocoagulation step. We tested the process with direct contact membrane distillation, but then we also got interested in looking at what would happen if we ran a crystallizer here and tried to run this to zero liquid discharge. People talk about the possibility of using a crystallizer and membrane distillation to go to zero liquid discharge. So that's some of the work we did. Excuse me. So electrocoagulation, electrolysis process, right? We have an anode and a cathode. We used aluminium. If this were in DI water, you would get oxidation at the anode. Aluminium ions would produce, then start to form these polyaluminium hydroxides. They will eventually start to coagulate and absorb organic compounds that are present in the produced water, which is what we're trying to remove, because these organic compounds can foul the membrane, because membrane distillation uses a hydrophobic membrane, putting it simply. Okay? And these flux initially are not very dense, so they float to the top, and then they will densify and sink to the bottom. At the cathode, you will get production of hydrogen if you're reducing water. However, that's in DI water. It's not quite as simple as this. So this is an example of our produced water. You can see the TDS is very, very high, right? 134,000, much higher than seawater. Total organic carbon is high, and that's what's causing fouling of the membrane distillation membrane. But you can see in this case, the major ions that are present, and actually most of it happens to be, where is sodium? Most of it is actually sodium, okay, sodium and chloride. But it could be some other things as well, calcium and so forth. And so one can work out the half-cell reduction potentials, and that's very important because one needs to know exactly how effective the electrocoagulation process is going to be, okay? So we did work on this, and then we did settling tests. So we looked at aluminium electrodes, iron aluminium, and iron electrodes, and we looked at basically the change in the liquid height, the interface with time, and this gives us basically a dimensionless sludge interface. The main point here is that actually the metal ion we use is very important. Aluminium leads to more open flocks, and they take much longer to settle, as you can see from the red line. If you use iron given by the blue, they settle much more quickly. The other point, I think it's well known, is that if you use too much flocculant, you can restabilize everything. So if you use too high a current, you will produce too many ions, if you like, and you restabilize it. So at 5 amps, actually the aluminium flocks take much longer to settle. Okay? So we did a lot of this work, but what was most important for us was removal of total organic carbon. And it turns out that aluminium was much more effective at reducing the TOC in the water after electrocoagulation. That's important because that is what is going to foul the membrane. Okay? And then we looked at particle size distribution, looked at flock size. Aluminium flocks again are much larger, they're more difficult to settle, but leads to better reduction of TOC. So anyway, this is the system we designed. This is at Texas Tech, based on these initial experiments and testing. And so we had basically it's supposed to treat 1,000 gallons a day. So here is our feed tank. We pump from the feed tank into our electrocoagulation reactor, which we designed. This is the EC reactor. There are a number of aluminium plates here. And then after electrocoagulation, the water flows into this other area here where it's held, and the residence time is about 25 minutes. That is important because it takes time. You need flock aging. It takes time for the polymerization reactions to occur. From there, it goes into a holding tank and then into these two tanks, that's these blue ones here, where we have ceramic submerged membranes. So our microfiltration or ultrafiltration step was using these submerged membranes, and then the permeate from that was the feed to the MD tank, to the membrane distillation tank. Okay. There are lots of other control systems here. We also needed a way to clean the electrodes because, of course, it's a corrosion process, right? You're destroying the electrodes, you've got to replace them. And so this is what we ran, and it was run for about a week and ran successfully. From our perspective, success meant that the membrane distillation process ran without fouling the membrane. Okay? And in the last 2 minutes left, so this is the process again schematically. We used DCMD, that was VMD, and we were interested in what would happen if you try to cool the feed reservoir here so you could try and run a crystallization process. Okay? So I'm just going to then jump ahead and say that basically if you do that, what you find is, if you look at the flux, this is the flux, say, for membrane distillation, and this is the flux for membrane distillation and crystallization. It's not that much higher. So while in the literature there's a lot of work that suggests that membrane distillation and crystallization could be viable, there are a number of problems with it. The first is, of course, it's much more energy intensive, right? A crystallizer takes up much more energy. Secondly, it is really not that easy to run because you don't want any sort of scaling on your MD membrane. You want all the scaling to be in the feed tank and then run this. And you can try to do that, but it turns out to be quite tricky. And actually, in terms of water recovery, we were not able to recover a huge amount more water. What we could do, though, is we could look at the precipitate that we formed, and we found that in this case we got pretty pure sodium chloride, which was expected because that's what was in there. And so this may be a way to actually mine the sludge if you want to, if that was of value and there were valuable components there. Okay, so I think I am just over half an hour, so I better stop. So basically, the conclusions from this work are that we developed a system to treat hydraulic fracturing produced water. This water is particularly challenging to treat. It met the specifications for the project. That does not mean it's necessarily commercially viable, but it met the required targets. And electrocoagulation, though, could be a very viable pre-treatment for membrane distillation and other sort of these sort of water treatment applications. Okay, and optimization of electrocoagulation is essential. I'd like to acknowledge funding for this work that comes from various sources, and then thank you very much for your attention.
Thank you very much for a very comprehensive and talk. Is there any question from the audience? Sure.
Thank you, Professor. Just wondering, your MEMD, membrane distillation, so this is the scale up, you have a pilot, or this is for research purpose?
This was for 1,000,
Yeah.
Basically the DOE wanted what they call a pilot, so it's 1,000, it was supposed to be 1,000 gallons per day.
Wow, it's 1,000 gallons per day.
Yeah, but
almost pilot, yeah?
Yeah, but I was going to say that the one point was that we were not supposed to do any membrane development. I didn't talk about that here. So it was supposed to be all commercially available membranes. Okay. And so actually my colleague who did this used a PVDF, commercially available PVDF.
So any like wetting issue or any because you use the MDC and MD.
MDC is only my work. That's an
Okay.
additional part. The pilot was simply, if I can go back to that
Yeah.
figure here, was oops, sorry, going too far. Was simply this. Yeah, that's the process. So this is the part we worked on, and then they had their MD step here. There was no idea of crystallization. Okay, that was something that we worked on later on. And so, no, there was no wetting issue for them. That was part of the whole thing, that we needed to make sure that our pre-treatment was such that they wouldn't have wetting of their membrane for the time that they ran it.
So no wetting. How long you have operate for this?
1 week. We
1
ran,
week.
1 week was the test.
Okay.
Now the system is all at Texas Tech, and they may be running it more, I don't know. But the test that we ran was for 5 days. 5 days.
5 days.
And the way we ran it actually, so it's not truly a continuous process, because the way we ran it was that we'd run our EC process, and then we'd collect water in the MD feed tank, and actually our EC ran much faster than the MD part of it. And then we have to stop, because after about 8 hours we have to stop, replace some electrodes, clean, and so forth. The electrodes are run such that the anode and cathode are only on the 2 outer electrodes, and so there's a differential rate of electrolysis occurring within the plates. Okay? But you can buy commercially available EC systems today that people are selling. And so, yeah. Yes.
Okay. Okay, let me continue the question that Professor Sean asked. So on MD, what was the TDS total dissolve salt going into the MD, and then after MD, the discharge from MD, what the.
So this bit I have to look up. I have forgotten what our limits were for TDS going into MD. It's in the publication, and you check that. But we had set these limits based on experimental results and the stability of the MD membrane. The discharge is something Marty did actually. And so I am not exactly sure what levels he got to at the very end of it, but I will tell you later. I can check the paper.
Oh, okay, okay. So essentially, okay. What I try to ask is how much amount of water that you can recover from the MD, from your original free.
Oh,
Yeah,
okay.
that's-
That's a very good
that's-
question. Yeah.
That's related to the total dissolve.
I agree.
And
It wasn't, as I, and again I look at the paper because I didn't really do that part, because I
Okay.
remember it was not that high. So
Okay.
maybe about 40% or something like this. It wasn't huge.
Okay.
It wasn't about 50%. I know that for
Okay,
sure.
what was the water in the MD? The temperature. What was the temperature?
Oh, he
then
was
again,
running
yeah.
60 was
About 60 degrees.
Yeah.
And was that already high enough? To give you enough? Then you.
But
then-
he's using VMD anyway. He's using vacuum.
Oh,
so
okay.
the temperature wasn't that critical, right? 'Cause
Okay,
he's using
okay.
vacuum.
You are using vacuum, then that's gonna add the cost, okay?
Yes, of course.
For,
Yes,
for your
yes,
vacuum.
yes.
Was there any falling problem for the PVDF?
Yes,
membrane?
yes, that's why a lot of our work on the upside pre-treatment was very important, because yes, PVDF does foul. And so it was, okay, there's a few more things. There's another problem, and that is that the quality of the produced water varies a lot.
Okay.
And so again, if you got some, and it really varied a lot. So sometimes you got stuff that was over 200,000 ppm starting, that cannot be treated. It's almost impossible by this
Okay.
process.
Alright,
So that's why the ones I showed you, which was about 100,000, we decided that that would be the maximum that we would treat. And then the process we developed with all the conditions that are in the paper will work for 100,000 ppm or less.
Okay.
But more than that, it won't. You will get fouling of the membrane.
Okay, all right. Let me continue on the following issue, because I did see this can be a critical, problem. So, for your other bio application, including virus,
Yes.
filtration
Yes.
and all.
Yes.
Okay, so the following can be a critical issue.
It's very critical for biotech. Critical.
Okay,
yes.
so for the case,
Yeah.
will a truly hydrophilic membrane help?
Yes, hydrophilic membrane will help, definitely. And extremely hydrophilic will also help.
Okay.
Because often, but it should help because often the denatured or misfolded proteins are more hydrophobic because the inside, which is hydrophobic, tends to be exposed to the surface. So typically, hydrophobic interaction chromatography, where you're trying to absorb, that is actually quite effective for this reason. That's why I showed in this, and it typically can help. In going back here, right at the very start, I had this slide. I didn't talk about it that much, but we used two different membranes. You see, we used here, this is the one here, HIC and ion exchange. So HIC does help. In this case, it didn't help quite as much as ion exchange. But hydrophobic interaction, so the idea is you're using a membrane that has got typically long chain hydrocarbons on it that are used to absorb more hydrophobic components, typically. That's how HIC is used. And that did help, but in this case though, it was ion exchange that was more successful. But yes.
Okay, you mentioned about cellulose membrane.
Yes.
Is that the most hydrophilic membrane being used
Yes.
for the application here?
Yes.
Have you tried for other more hydrophilic?
We have not. We have not.
Okay.
We have not.
Okay. The reason I'm raising this is the most hydrophilic membrane that I know is the porous mega.
Yeah.
Okay, that's essentially the glass.
Right.
Extremely hydro.
Right.
Have anyone used that?
I am not sure. I haven't seen anything on that.
Okay, so for that application, particularly you can control the pore size
Yes.
of micromembrane very precisely.
Right.
This is using the irradiation
Right.
etching process,
Right.
and you can get down the pore size down to the smallest one, if I remember correctly, somewhere in the order of about 50 angstrom.
Okay.
Okay, and then you can make it bigger too.
Right.
Okay,
50
you know
angstrom is too
etching
small.
process.
You need bigger. Yeah.
Okay, so do you know anyone have used
I
that?
don't know anyone who has used mica.
Okay,
No.
so in this case, it can be a very selective separation.
Yes.
Cannot be for this large scale. For large scale, for your produced water.
Right.
No way. Yeah, you cannot. Yeah.
Right. But this is much smaller scale.
Yeah,
This is
for
much
the
smaller.
small scale,
Much
but more really for extremely selective you really need extremely hydrophilic membrane,
Right.
particularly for the medical application.
Right.
That can be.
But mica, yeah, you're right. You're right. I don't know of anyone who has used mica in biotech.
Okay.
At least.
But I think for the
not-
medical application, I believe that can be a
Yeah.
very effective membrane.
Yeah. There are two other things. You also talk about, I was telling about cell therapy. So people are looking at like personalized medicine. So you take your own cells and you grow them somehow and use them to treat cancers and things. So then you talk about very small volumes, right?
Okay.
Whereas this is still relatively large scale compared to that. You're talking about 10,000 liters or sometimes more than that in these applications. But
Okay, for the larger scale, for the practical application, how do you deal with the volume problem? So you just need to turn up to operate for a period of time, flux decline so rapidly, then you stop, right? And then you turn the membrane,
yeah.
and then reuse again.
So in batch operation, for ultrafiltration and also for bioreactor harvesting microfiltration, yes. But you don't stop in a batch. You need to be designing this for it to be successful. You run the batch, then you stop, then you regenerate. You cannot stop in the middle of a batch.
No, I'm saying that you will be switching, okay? There will be a switching.
Yes, you can do that. For virus filtration, you do not regenerate. You run and throw away.
Okay.
It's one-time use. Because you will never be able to validate that you removed any virus particles. You can't validate that. So again, if you have flux decline like this is not going to work. This will not work. And so you either have to come up with a different system, different membrane, different buffer conditions, different operating conditions, or larger surface area, or you're going to do something.
Okay.
I think we have time for one last quick question. Any students? Yeah, Sam?
Thank you very much. Sorry. Thank you very much again. Amazing presentation. I'm just wondering, even though in time initial flu, virus or microorganisms, when they subjected to the membrane surface shear rate, the structure will be damaged or not?
It's a good question. Yes, they can be. If you think of specific proteins, if you're trying to produce a monoclonal antibody, then yes, they could be shear sensitive. So you have to be very careful about the shear rates you use. And that gets back to this whole idea of trying to do continuous biomanufacturing, because that would be actually much more gentle. If you have the protein coming in, going through because feed pump actually causes a lot of shear. Going through that once, entering the module once, then the rest should be uniform, and then it comes out and you collect it, it goes to the next process. If you are running around in a circle, you keep going back to the feed reservoir, back and then back, that leads to much more. So that's one very good point. And so, yes, this is very important. Viruses, so I only talked about AAV and lentivirus briefly, and these are so-called non-enveloped viruses. They're fairly rigid. So some viruses are very good. They are rigid, they're strong. So they are relatively not shear sensitive. There are others that are very shear sensitive, and so processing them by any sort of flow process like this would be very difficult. So if you want to discuss more about viruses used for gene therapy applications, yes, there's a whole range of them, and some are really not practical because they might be very good for delivering your product, but they're not very processable, you see. And so there are many examples of this. You know, people talk about using measles. I don't know if you know this measles virus. It's a very interesting one, because evidently it's a very infective virus, and it has been shown that it could be a cure for leukemia because it's very effective at quickly going and attacking highly cells that are reproducing very rapidly. And so if you are able to inject it and do this and then give the patient immunity to measles, most of us have got these days measles vaccines, so you'd have to reduce the immunity then bring it up again. It could be very effective, but it's almost impossible to process because it's very, very fragile. So yes.
May I have just a short one? What about if we try to immobilize zwitter ionic mole on the membrane?
Yes.
Do you think, based on the hydration bound, it can decrease the shear rate or not?
Decrease shear rate, I'm not sure. You can certainly try to decrease fouling, right? If you're trying to increase hydration there. How does it decrease shear rate? I'm not quite sure.
When you immobilize ions,
Yeah.
the bounded layer, water bound layer thickness increases. Then viruses can go through on a, how can I say, on a pillow of the water.
Yeah, I'm not sure that that's going to be.
I noticed that somebody in
Yeah.
the USA writes some such kind of concept. I saw thinking.
Yeah, I know her. Yeah. But I think for this particular application, that would be difficult. That would be very
Oh, yeah,
difficult.
definitely.
Very difficult. And I'm not sure how much you would really gain from it. Yeah.
Well, thank you.
Okay, let's thank Professor Wickramasinghe.